Today's lecture is on eukaryotic cells. So here's just a chart that shows you some differences between eukaryotes and prokaryotes, and one of the big things is that eukaryotic cells do have a nucleus, prokaryotes don't, and the ribosomes are 80S in eukaryotes, and prokaryotes only have a 70S ribosome. So again, big things. Eukaryotic cells have a defined nucleus. Now they can bring in things into their cells in two ways. One, endocytosis, which is importing things into the cell, and then exocytosis, which is exporting things out of the cell. So two ways to transport. So during endocytosis, the plasma membrane is going to fold in and then pinch off and form what are called endocytic vesicles. And there's three different types of endocytosis: pinocytosis, phagocytosis, and receptor-mediated. But pinocytosis and phagocytosis are what I would like you guys to know. So pinocytosis is ingestion of liquids and small molecules. And phagocytosis is ingestion of large molecules and cells. So for your exam, I'd like you guys to know the steps of phagocytosis. So the first thing is that a non-dissolved target, like let's say a cell or virus, is engulfed, okay? And then we get what's called a phagosome that will form. Then the next thing that happens is the phagosome is going to fuse with a lysosome, and it's going to form what's called a phagolysosome. Then hydrolytic enzymes are going to destroy the contents, so the microbe, for instance, inside, and then eventually anything that's not digested is going to be released. So please know these 5 steps of phagocytosis. So again, there's a lot of differences here. If we look at the eukaryotes, we have animals, plants, fungi, protista, etc., and they have a lot of interesting features. So as far as animals go, the main one I want you guys to know would be parasitic worms. Please know that those are considered helminths. And they have very complex life cycles. And it's interesting, guys, because World Health Organization estimates half the world's population is infected with some type of a helminth. Fungi, there's about 600,000 different species. Most are multicellular. Yeast, however, happen to be unicellular. Now, most fungi grow as a collection of structures that we call hyphae. So hyphae are filaments. Please know there's 2 types of hyphae. Septate, they have divisions between the cells. And we have aseptate, or coenocytic hyphae. They don't have those divisions between the cells. So again, here's septate. You can see the walls in between, and aseptate or coenocytic, these are just a continuum without the walls in between. Now, dimorphic fungi can be mold-like or yeast-like depending on the environment, and that's an important fact to know because a lot of pathogenic fungi are dimorphic, and pathogenic means those that cause a disease. So mycology then would be the study of fungi, and a mycosis would be any fungal infection. So most of the fungal infections will occur in immunocompromised people, for instance, like AIDS patients. And sometimes what happens is that if you disrupt your normal microbiota, let's say you're on a lot of antibiotics, a yeast infection might develop as a result of that. So one type of fungal infection I'd like you guys to know is caused by dermatophytes. These are 2 pathogens. They will infect your skin, hair, and nails. And here's an example of toenail that is actually being eaten away by fungus. And on this side, this is actually a really grown up or blown up example of some different fungi that are actually on someone's foot. Now, protists are unicellular, multicellular, multinucleated. They can have sexual or asexual reproduction. Sometimes they have a cell wall, sometimes they do not. The one group I'd like you to be aware of are called apicocomplexa. They're the largest phyla of protozoans. Within that group, I'd like you guys to be aware of the one that causes malaria. So please know, malaria is caused by Plasmodium. We're going to be talking about toxoplasmosis later, cryptosporidium, and that kind of thing, but for now, just know, malaria is caused by Plasmodium. Now, the eukaryotes that do have cell walls would be things like fungi, plants, and some of the protists, but they're a lot different than bacterial cell walls. Cilia, I'd like you to know, are structurally similar to flagella, but they're much shorter. And cells that do have those, normally there's a lot more cilia than you would see flagella on cells. Please take a screenshot of this slide and upload it to the Dropbox. Ribosomes in eukaryotes have 70S size, okay? Eukaryotes have a 70S ribosome. Again, eukaryotes have a nucleus, also endoplasmic reticulum. They have a rough and a smooth endoplasmic reticulum. Golgi apparatus, there's a lot of different transport that takes place with the Golgi. It kind of comes right off of the endoplasmic reticulum. And please take another screenshot and upload it to the Dropbox of this one. And for this one, guys, we're looking at vesicles and vacuoles. And I want you to be aware of the fact that lysosomes will contain hydrolytic enzymes that break down substances engulfed by a cell. So for this chapter, guys, please note, there's 2 screenshots I'd like you to upload. And I'm just trying to make sure you're going through all of these slides. Mitochondria, we'll talk a lot about electron transport later, and they're kind of like the powerhouse, if you will, of the cell. A lot of ATP production takes place there. Chloroplasts are not found in animal cells, however, they are found in plants, and that's where your photosynthesis takes place in a plant.